I would like to thank the chairs and ASTMH. We skip past the title. So the gold standard for vaccine development is the smallpox eradication program. The smallpox vaccine consists of attenuated virus delivered by skin scarification with a bifurcated needle. For our studies, we use synthetic peptides, which are awfully poorly immunogenic by themselves because they are highly purified and they lack pathogen-associated molecular patterns. These molecular patterns are recognized by toll-like receptors, TLRs, that are located on the outside of cell membranes and within the endosome. And synthetic TLR agonists can serve as adjuvants to initiate the innate immunity and boost adaptive immune response. So our lab has been using p-falciparum CS repeat peptide mixed with risdiplamide, a small synthetic molecule that is recognized by TLR7 and 8, and CpG, a bacterial DNA motif recognized by TLR9. These components are mixed with Ativax, a squalene oil emulsion for delivery. For our study, we immunize mice by skin scarification on the dorsal region of the back, and we collect sera at 14 days post each immunization, which is indicated here by the arrows. ELISA is used to screen for anti-IgG, anti-CS IgG antibodies, and skin scarification with peptide plus PBS alone gave fairly modest IgG endpoint titers. The endpoint titers are on the log scale here. And in contrast, peptide plus Ativax gave threefold higher IgG antibody titers, and peptide plus Ativax with a combination of TLR7 and 8 gave sevenfold higher endpoint titers greater than Ativax. We further screened the pooled immune sera to determine IgG subtypes, and we used IgG2C as a marker for a Th1 type immune response and IgG1 as a marker for Th2 type immune response. The groups that received peptide alone in PBS by skin scarification or with Ativax were predominantly an IgG1 Th2 type response, whereas the shift to IgG1 with a IgG2C with a Th1 response occurred with the addition of TLR agonists. Similar results were achieved in the LE spot with T cells from mice immunized with Ativax, peptide plus Ativax or peptide plus PBS, producing IL-5, which signifies a Th2 type response, and the group that received Ativax plus TLRs produced predominantly an interferon gamma type response. These cells were, the cytokine-producing cells were CD4 positive T cells, as the spot-forming cells were depleted with the addition of monoclonal antibody CD4, but not with anti-CD8. To measure the neutralizing neutralizing antibodies in the sera, we used a transgenic rodent model that expresses p-falciparum CS repeats. And for this, we incubated in vitro immune sera or monoclonal antibodies with sporozoites, transferred these to cell culture plates with human hepatoma cells, and after 40 hours incubation, extracted RNA and quantified levels of pBurg18 sRNA by real-time PCR. We found a dose-dependent neutralizing antibody response in the group that received TLR agonist plus risdiplamide with a 1 to 10 and a 1 to 20 dilution greater than 90%. The line here is signifying 90% cutoff and reduction of parasite invasion to HepG2 cells, and we're at a similar level to monoclonal antibody 2A10, which serves as a positive control. In contrast, the groups that received peptide plus Ativax or peptide and PBS were not significantly reduced and had similar levels of 18S RNA as the naive control mice. To measure protection in vivo, we challenged immunized mice with approximately 10 infected mosquitoes that are infected with transgenic parasite, allowed them to take a blood meal, and 40 hours later, we harvested the liver, homogenized the tissue, extracted RNA, and quantified 18S rRNA by qPCR. Similarly, we found that the group that received peptide plus Adavax in the combination of resiquimod and CpG had the greatest reduction in parasite burden as compared to the lack of protection found in the peptide plus Adavax or PBS groups. So to understand what was giving this protective adaptive immunity, we wanted to look at earlier time points to look at the innate immune response. And shown here at 4 hours and 24 hours post-skin scarification. And shown here are H&E stains of skin tissue sections. At 4 hours, skin scarification with peptide plus PBS is only noticeable as a break in the epidermis. Similarly, the group that received peptide plus Adavax and resiquimod and CpG had similar morphology. In contrast to 24 hours, the skin scarification site is readily visible with ongoing wound repair. And with the peptide plus Adavax group or peptide plus Adavax and TLR combo, there's cellular infiltration through the subcutaneous layers and dermis. To characterize the cell infiltrate, we used immunohistochemistry at the 24-hour time period based on the H&E staining. And staining with antibody F480 to recognize macrophages. The group that received peptide plus PBS did not show significant levels of, substantial levels of positively stained cells with F480. The group that had peptide plus Adavax showed an increase in positively stained cells for F480. And in contrast, the group that received peptide Adavax and resiquimod CpG combo did not show many positively stained cells associated with the skin scarification site. When staining with CD11b MAC1 for staining of monocytes and macrophages, all three conditions showed heavy staining of positive cells associated with the skin scarification site, and apparently more so with the peptide plus Adavax group. We did some further staining with antibody for CD4 for T cells and antibody B220 for B cells at the 24-hour time period. We found CD4 positively stained T cells, or positively stained cells in the dermis and associated with hair follicles, but there weren't any signs of CD4 positively stained cells with the cell infiltrate. We also stayed with anti-CD3 antibody and found similar results. The staining with B220 was also negative with no association with positively stained cells with the cell infiltrate. To measure cytokine and chemokine levels at the earlier time points, we utilized Ray Biotech cytokine microarray, which is essentially a sandwich-based ELISA where an array of antibodies are coated on a glass slide. And using a standard curve, we're able to quantitate levels of cytokine and chemokines within the unknown samples, in our serum samples in this case. And the panel here in this microarray has 18 different cytokines. So the microarray showed us that we had IL-6, IL-22, IL-17F, and MIP3A, all in the group that received skin scarification with CS peptide, Adavax, resiquimod, and CpG, whereas the groups that did not have the TLR agonists were negative and below the line of limit of detection of the assay shown in the red line. For IL-6, 22, and MIP3A, these levels were significantly higher at 4 hours, but they were diminished by 24 hours, while the IL-17F persisted at 24 hours. In the second serological assay, we used a CBA to screen the sera at 4 hours, and again we found IL-6 to be IL-6 to be present as we did in the microarray, but as well we found IL-12, TNF-alpha, and MCP-1, again in the group that received peptide, AddaVax, and TLR combos. And when we immunized the mice with single TLRs, AddaVax plus resiquimod or CpG, we were able to determine that the resiquimod seemed to be the agonist that was stimulating this innate response. So beyond that series of cytokines, we also were interested in IP-10, which is an important chemokine for attracting cells in the skin. And we were able to detect this by a standard ELISA at 4 and 24 hours post-skin scarification in the group that received CS peptide plus AddaVax and resiquimod plus CpG at the 4 and 24-hour time point, while we didn't detect it in the other groups. So in all, we were able to use 3 different serological assays, the microarray, CBA, and the standard ELISA to detect 8 different cytokines and chemokines that are of inflammatory type response at 4 hours post-skin scarification. So to summarize, skin scarification with p-falciparum CS peptide and AddaVax plus TLR7, 8, and 9 agonists can elicit protective immunity. Protection is characterized by the induction of high levels of anti-repeat antibodies and a Th1 type immune response. The antibodies elicited by skin scarification immunization are functional and inhibit sporozoites both in vitro and following challenge in vivo. The chemokines and cytokines detected as early as 4 hours post-skin scarification predict the induction of the sporozoite neutralizing antibody in the Th1 type adaptive immune response. So skin scarification provides a facile method of screening vaccine adjuvants for further clinical development and optimization of delivery systems such as microneedles. So I would like to say thank you to the Narden Lab with Rita Altier and Sandra Gonzalez, who runs our parasite cycle, and the Freevert Lab, Rashawn Johnson, and our funding. Any questions?
Comments, questions? So, Robert, I wanted to know, did you look for CD4 cells in any draining lymph nodes that would drain the site of the scarification?
For the hyperimmunized mice?
Right. Since you didn't see any in the skin.
We didn't show it, but we did find a response.
So you found them in
it was at lower levels of cytokine production, but yes, it's there. Okay. We also realized this is a mouse model, and we have ongoing studies with a porcine model for the skin of the pig more closely resemble human skin, and we can also test their sera in the neutralization assay.
Okay, thank you.